Hello everyone, welcome you all in this video tutorial. So in this video lecture, we are going to discuss about non-coding DNA sequences or introns. So let's start with the introductory part. So in this video, we are going to discuss about non-coding DNA sequences. So before moving to the topic, it's very important to discuss some basic facts about what is DNA and what are the different types of DNA and DNA sequences. Now the DNA is also called as deoxyribonucleic acid and it is a type of nucleic acid. We know that nucleic acids are the most important macromolecules found in all the cellular organisms, and there are two types of nucleic acids which were first discovered by Frederick Misser. So the first one is DNA named as deoxyribonucleic acid and the another one is RNA also named as ribonucleic acid. Now, DNA acts as a hereditary material in all the organisms, except some viruses and some prokaryotic organisms, where the RNA molecule acts as a hereditary material. The hereditary material means the DNA molecules or the RNA molecules are responsible factors to inherit the parental traits into their offsprings. Now, there are two types of cells. The first one is prokaryotic and the another one is eukaryotic cell. So in the case of prokaryotes, DNA found in the cytoplasm of the cell because there is not well-defined nucleus in these organisms. But in case of eukaryotes or eukaryotic cells, DNA occur in the nucleus of the cell because here the cell consists a special organelle which is covered by nuclear membrane and called as nucleus. So in the nucleus, DNA found in a compact structure called as chromosome, and chromosome has found in the nucleus which is made up of DNA and some histone and non-histone proteins. So here the DNA helps in different types of cellular activities like replication, transcription and many other cellular activities. Now the DNA is made up of nucleotide sequences and made up of genes, because these genes are considered as the unit of heredity for all the cellular organisms. So the DNA have two types of sequences which are named as coding DNA sequences and non-coding DNA sequences. So in this video series, we are going to discuss about the non-coding DNA sequences also as introns. So let's discuss about detail about the non-coding DNA sequences or introns. So basically the introns are the stretches of non-coding DNA which are found in genes between the exons or coding DNA sequences. Now the non-coding DNA sequences have not ability to code for amino acids. We know that DNA perform all the cellular activities with the help of proteins. So by the process of translation, the DNA script converted into RNA and then proteins, and these proteins are the macromolecules which are responsible for all the cellular activities because they are perform as enzymes. All the enzymes are proteinous in nature except some ribosomal enzymes, and also proteins are the building blocks for muscles and all the connective tissues and many other cellular organelles. So the non-coding DNA have not ability to code for the amino acids or proteins, and these sequences lies between genes on the chromosome between the exons. Now they have not any known functions because they have not ability to translate. But there are some non-coding DNA molecules which seen as play a role in regulation of gene expression like promoters or many other functions. So here in this diagrammatic presentation, we can see the location of introns in a particular gene. So this is the DNA stretch or a double helical DNA structure. So in this DNA structure, we can see different introgenic species, and in between these intergenic species, there are genes located in the DNA molecule. So when we see a structure of gene in a detail, so this is the structure of gene which is made up of introns and exons. So here in this gene, we can see the blue colored regions which are denoting the introns or non-coding DNA sequences, and in between these introns there are orange colored regions which are resembling the exons or coding DNA sequences which are capable for the protein synthesis with the process of translation. So let's discuss about the discovery of introns. So the non-coding sequences or introns was first discovered in 1977 with introduction of DNA sequencing. We know that what is the DNA sequencing? It is the process of sequencing of all the genes present in the DNA. So when the DNA sequencing arrived, it was initially believed that the mRNA sequences or the mRNA script was simply trimmed at the ends, because it was seen that after the process of RNA splicing, the mRNA script shortened in its length. But after the study about DNA sequencing, it was revealed that most of the removed transcript come from the internal regions, which revealed that there are some introns or some non-capable for protein synthesis, and these regions are removed by the process of RNA splicing. The term exons and introns was first coined by Gilbert. So now let's understand about the structure of non-coding DNA and introns. So the introns are the sequences of DNA that starts and end with specific nucleotide sequences, and these sequences act as the boundary between introns and exons. And they are also called as splicing sites because these sites get spliced by the process of RNA splicing. So in the case of humans, most of the introns begins with 5' end by a sequence GUA and end at the 3' end by the sequence CAG. So here in this diagrammatic presentation, we can see the structure of introns and also gene, and also see the location of introns. So this is the DNA stretch having introns and exons in its structure. So when this DNA molecule is get transcribed, it is converted into mRNA script. So this is the mRNA script, and it have both exons and introns. So basically this mRNA script is spliced by the process of RNA splicing, because these introns can affect the process of protein synthesis, because they have not ability to get synthesized or to get translated. So these introns are spliced or removed by the mRNA script, and then the proper mRNA script is transported to cytoplasm, where this mRNA script is get converted into protein molecules by the process of translation. So now let's understand about the functions of non-coding DNA sequences. So basically, initially it was considered that they are a junk DNA sequences and they have not any essential role in the cell because they are removed by the process of RNA splicing. But later it has been shown that introns play an important role in regulation and gene expression, because we know that they are only present in eukaryotic organisms, and in these organisms they have an important role to increase the length of the gene, because they found in between two exons and 90% of the genes are introns. So in that condition, they increase the chances of crossing over and recombination between sister chromosomes, and we know that the process of crossing over enhances the chances of mutations. Now the non-coding DNA sequences also help in alternate splicing, We know that what is the alternate splicing? It is the process, and this process allows a single gene to encode for multiple proteins because they can be assembled in multiple ways. When introns get spliced or removed by the process of RNA splicing, the exons can assemble in multiple ways, and that condition they can code for different proteins. So these are the functions of non-coding sequences in organisms.